And I am very excited to have everyone on this particular chat, as it is a subject that I have been following for quite some time. And we got a very amazing person, probably one of the best I've known in the field for the last, well, about 5-6 years at explaining what's happening in studies, science, and a great breakdown. So we have Gail Broder from the HIV Vaccine Trials Network. She's going to give you guys an overview of what the latest AMP study is, which is scientifically a really amazing breakthrough, potentially, and has some real implications on the direction of HIV science and prevention and where we're going as a field, and it's a great timely topic for people to know about. So just some housekeeping. Remember the Brown Bag Lunch series. We'll have one more brown bag in December. I'll be sending out the information on that probably next week, and will be about long-acting injectables for both prevention and treatment. So look forward to that. But also, for those who haven't yet done so, please fill out the survey about Brown Bag Lunch so that we can set the Brown Bag calendar for next year and continue giving you great webinar activities to learn and teach people back in your own regions. Just some housekeeping. If you have any questions throughout the presentation, please ask them within the question box. We'll be able to log and track those. At the end of the presentation, we'll do an open question period. If you want to ask a verbal question, please hit the raise hand button. It's a little hand that's shown there, and I will unmute your line, and you'll be able to ask your question directly. And with that, I'll hand things over to Gail.
Great. Thank you, Matthew. Hope everyone can hear me okay. Please let me know if it gets quieter or if I need to change anything. As you can see, we're talking today about a new study, HVTN 703, HPTN 081, which is a whole lot of letters and numbers to say in ordinary conversation. So we have dubbed it the AMP study, and I'll be talking today a little bit to tell you what AMP means and what it stands for. So who's doing this study? This is the first time we are doing a collaborative trial between two of the prevention networks that are sponsored by the National Institute of Allergy and Infectious Disease, both the HVTN that I'm a part of and the HIV Prevention Trials Network, the HPTN. Some of you may know my HPTN counterpart on this trial, Jonathan Lucas, who is coordinating all of the community engagement activities on the HPTN side of things. Next slide, please. To give you a sense of where this trial is going to be happening, we have two different cohorts that I'll be telling you about. One cohort will be in the Americas, the U.S. and in South America. The second cohort is in Africa, primarily Eastern and Southern Africa, as you can see from the dots. And since this is a group that's focused in the U.S., on the next slide you can see the breakdown of our U.S. cities, if you would be so kind. Thank you, Matthew. So you can see where we will be. And in a couple of the cities we have multiple locations. In Atlanta we are at both the Hope Clinic and the Ponce de Leon Clinic. In Boston, we're at Fenway and at Brigham and Women's Hospital. And in New York, we will be in 4 locations, at Columbia University, in the Physicians and Surgeons building up in Washington Heights, in Harlem, in the Bronx, and at the New York Blood Center at Union Square. And the rest of the cities have just 1 location apiece. Next, please. So I mentioned we have 2 cohorts. In Africa, we'll be working in heterosexual women, and in the Americas, we'll be working with men and transgender individuals who have sex with men. In both cohorts, we recognize that these are populations that continue to be disproportionately impacted by the epidemic, and they're not currently being well served by other prevention methods that we have available. Although with the approval of PrEP, we hope that will continue to be taken up and getting into the communities that need it most. But what we really wanted to be sure of was that as we work to develop any kind of new prevention methods and new options for folks, that the people who stand to benefit the most from those new methods are involved in the trials and involved in determining whether or not they may work. So that's really the focus behind why we've selected these populations to participate. I also want to note that with regard to transgender men, also known as people who are female to male, this is a group of people that has previously never been included in an efficacy trial of any kind of HIV prevention strategy. So we're very proud that we've been able to include them as a named eligible population. Next, please. So this is, I'm sure, a metaphor that you are all quite familiar with, the toolbox. And obviously we have lots of things that work, but if they were sufficient, the epidemic would not be where we are today. So our goal is to continue to add tools to that toolbox, and that's really the focus of both of the HPTN and the VTN, to continue to find new methods to add to the toolbox to continue to try to meet the needs of a wide range of people in a wide range of circumstances around the globe. And obviously, as you can see from the different color, we don't yet have microbicides or vaccines. Those are still in the research stages. And as we get through this presentation, hopefully I'll be able to help you understand why this new study helps to inform the vaccine field. Next, please. So considering an analogy, one of the things that we have learned from the family planning movement in particular is that the more options for prevention that you have, the greatest opportunity there is for people to find an intervention that will work for them, particularly as their needs change over the course of their lifetimes, as circumstances vary even just month to month or year to year. So this slide gives you a little bit of a comparison between thinking about preventing pregnancy and thinking about preventing HIV. And obviously, we know that things like condoms work. We know that taking a daily birth control pill works, just as we know that taking daily Truvada works. We also have some sort of emergency interventions like morning after pills and post-exposure prophylaxis, or PEP. And we hope that we will have topical microbicides that would compare to use of spermicides. We hope that we'll be able to have long-acting interventions such as antibody-mediated prevention, which we're talking about today. And it looks like you'll be talking about the long-acting injectable PrEP in your future meetings. So that would be another comparison for long-acting birth control, like implantable birth control. And then the more sort of do it once and you're done sort of strategies like vasectomy and tubal ligation, which compare to vaccines, which are the longest acting prevention that we know of for any sort of infectious disease. And we hope that we'll have an HIV vaccine to add to that mix. Next, please. So in particular, when we think about all these different kinds of prevention options, we're really looking at what's missing, and where we need more options is around the times when people are potentially being exposed to HIV, and that's around times in particular associated with sex. So whether it's before, during, or after, those circled bits in the middle, we now know that we've got Truvada PrEP that works, and shown in italics are some of these other options that are being researched. But again, these are the focuses of our two networks, is to try to increase the options that would address this period of exposure and hopefully add to the prevention options that people have. Next, please. So how do people fight infections? And that can be a much more complicated answer, but one way, and the way that we'll focus on today, is by using antibodies. And antibodies are natural proteins that are made in our bodies. They're shaped a little bit like the letter Y, and you can see them in sort of neon green in this drawing. They are part of our immune system, which is what protects us against disease and illness. And what's interesting about antibodies is that they are specific for every different kind of illness or germ that we might ever get exposed to. So if you had a cold last winter or you had the flu that was circulating last year, you have particular antibodies for that type of cold and that type of flu. Similarly, if you had chickenpox as a child, if you had ever had measles, every different kind of infection, we have unique and specific antibodies to help us fight back. But what's interesting is that they can also be manufactured in a lab using the same kinds of controlled environments and procedures like we use for making medications that come in pill form. And so they can be used in the same sorts of ways like a medication. And in just the past few years, there have been several different research groups that have made discoveries about particular kinds of antibodies called broadly neutralizing antibodies. And you'll also hear that abbreviated as BNABs. Scientists love to have shorthand for anything that they can make it harder for lay people to follow in conversation. And what's interesting about these broadly neutralizing antibodies, these BNABs, is that they're rare and they take a long time to develop in the body, often more than 10 years to develop. And unfortunately, that's part of why they've been very difficult to utilize thus far. Because of the fact that they take so long to develop naturally, they're not particularly effective in preventing an HIV infection. But in some of the people that they have been discovered in, they are people who are long-term non-progressors. And the thought, at least, is that they may play a role in helping people control viral load and keeping viral load suppressed in a natural way. So the people who we think of as elite controllers, for example. And so now the strategies have turned to trying to figure out how we can take these BNABs that have been discovered naturally in real people and convert them over to something that can be produced in a laboratory, that can be purified and sterilized and made safe to use in other people. And that's been a big focus for the last several years. So if you would advance, Matthew. There we go. And there's actually been a rather long history of using antibodies to prevent particularly infections caused by viruses. So on this slide, just to give you a point of reference, these are other viruses listed down the left, and the different kinds of antibody-based products that we have that can be used to either prevent or use after exposure to help control disease. Perhaps the most relevant for us in the AMP study is the one second from the bottom. RSV is the abbreviation for respiratory syncytial virus, which is an infection in babies. It causes a lot of respiratory lung distress, and it can actually be deadly to infants. And there is an antibody called, I never can say it, palivizumab, that can be given to babies at risk to prevent that infection. And that is actually one of the specific antibodies that inspired this trial, because one of the hopes is that if this antibody is found to be effective, it could potentially be used in HIV-positive women who are pregnant to help prevent infection in their newborns and also to prevent further transmission through breastfeeding. So that disease gives us a particular model that we might follow that's relevant for HIV. Let's go ahead and keep going. So what is AMP, this acronym? AMP stands for Antibody-Mediated Prevention, the idea of using an antibody against HIV that was made in a laboratory, giving it to people through an intravenous infusion, which most of us know or have seen on medical TV shows. called an IV. And in Africa, it's often known in communities as getting a drip. This is the idea. Actually, I think for many of us, we associate getting IVs with treatment for illness like cancer and chemotherapy. But in fact, IVs can be used for healthy people as well. They're used for people who get dehydrated and need to receive fluids. They can be used to give vitamin infusions. So there are other uses of infusion besides just treatment of illness, even though that's typically how we see it portrayed in American culture. Let's keep going. So what is the antibody that we're testing in this particular study? In this study, the broadly neutralizing antibody, or BNAB, that we're using is one called VRC01. It was identified by the Vaccine Research Center, hence the VRC in its name. The VRC is part of the National Institutes of Health at the US NIH, and they have now actually identified several different antibodies. This is the first one that they identified, and it's the farthest along in terms of its development and study. It is unique because it attaches to HIV at the CD4 binding site. And this is the same spot that HIV would ordinarily use to attach onto our T cells to cause an infection. And so if you can sort of think about if your hand, or if you made a fist, if your fist was representative of the HIV virion, this antibody works if you want to sort of pantomime along with me. If you take your other hand and cover over the top of your fist so that you're covering over your knuckles and the tops of your fingers, that's how this antibody latches on. It covers over that surface area of HIV that would normally be used to cause an infection, and in doing so, it neutralizes HIV and it blocks it from being able to attach. This particular antibody has already been studied in several small phase 1 studies. It's been given to over 100 people, and so far it's been shown to be quite safe, and there have been very few side effects associated with it. The side effects that we've seen so far are typically associated with getting an IV and not necessarily with the antibody itself. So there are things that we've seen like getting a little irritation at the spot on your arm where the needle was inserted, or having that spot be tender and red afterwards. But we've had very few we've had no significant side effects, and we've also had none that are severe. The only side effects that have been reported have been very mild, and they all go away quite quickly on their own without any sort of treatment or intervention. So this study, the AMP study, will be the first time that we're using the antibody in a large group of people, looking specifically at the question of, can it prevent infections? This is the first time that research question will be addressed. Next slide, please. So just reviewing a little bit of terminology, we've mentioned that it's a broadly neutralizing antibody. It's also called a monoclonal antibody. So if any of you are word nerds the way I am, you can break that term monoclonal down a little bit. Mono meaning one, and clonal meaning like the word clone that we've heard of. Some of you may remember the cloning of sheep that happened in England. Cloning means to make copies. So we have this one antibody that we're making bajillions of copies of. As I mentioned, it was discovered by scientists at the US NIH. And so far in lab studies, and this is the reason that it's of such interest to take it into further human study, in lab studies it's been effective at neutralizing about 90% of the samples of HIV that it's been tested against. And in just a moment I'm going to show you a little more detail about that. And just to give you an idea of what it looks like, on the right in that drawing, the gray squiggles at the top are the GP120 with the protein spike on the surface of HIV. In red is the spot on that GP120 known as the CD4 binding site. And in green and purple, you can see there, that is the VRC01 antibody bound on, attached to that CD4 binding site. And you can sort of look at it, if you use your imagination. I mentioned that antibodies are sort of shaped like the letter Y, and you can sort of see there the green and purple make the two arms of the letter Y, and the green piece is a little longer at the bottom. That's kind of the tail of the antibody at the base, just to give you an idea of what it looks like. Next slide, please. Yes, this is the data about its neutralizing. So this, I know at first glance, looks like a scary science data slide, so hang in with me for a minute and I'll walk you through it. Those two diagrams at the top that look sort of like snowflakes in colors, that is VRC01 on the left, and on the right is B12, another antibody that we've known about for some time. And all of those little lines in the snowflake diagrams represent a different sample of HIV-infected blood that the antibody was tested against in a laboratory. And in total, those lines represent 190 different samples. You can also see in very tiny print around the little snowflake there are letters, and those represent the different clades of HIV represented in those blood samples. So you can see kind of to the upper left is clade B, which is what's common in the US. Down towards the bottom center, clade A. Over towards the right, clade C, which is common in sub-Saharan Africa. So you can see that it's been tested against a lot of different samples from around the world. And the colors give you an indication of how well the antibody was able to neutralize those particular samples. So in black, a sample that was not neutralized at all. In green, a sample that was only partially neutralized. It didn't really kill it off in its entirety. And in red, a sample that was completely neutralized, where the antibody was able to completely block HIV. And so just by looking at the color scheme, if nothing else, you can see on the left that VRC01 has a whole lot more red. It's a much more effective killer than by comparison to the B12 antibody shown on the right. And if you get down into the numbers below, you can see that overall, the VRC01 at a higher concentration was able to neutralize 91%. But even going down to a very small concentration, less than 1 microgram per milliliter, it was able to neutralize overall 72%. And then if you're interested in the particulars of how it performed against any individual clade, you can see those in the columns there. So clade B in the America, at the higher concentration, it neutralized 96%. At the low concentration, it neutralized 80%. Clade C in Africa at 87% and 66%, respectively. So this is the kind of data that has scientists so excited about this antibody. We've not seen anything else that is so successful at being able to neutralize HIV in lab samples and lab tests, and that's why there's been such excitement about bringing it into human studies. So next slide, please. So why do we want to look at it? We think that it has promise for HIV prevention. As I mentioned, we know that it's been very potent and able to neutralize broadly strains from around the world in lab studies. Our early phase studies in over 100 people have had great safety results. And we think that it may be something that could supplement other kinds of prevention approaches. And I can say more about that later if we have time. We also think that it has the potential to move the HIV vaccine search forward. One of the research questions that the study is going to answer is how much, if we find in fact that this antibody is successful at blocking HIV and neutralizing infection, We need to know how much antibody does somebody need to have in their system to achieve that protection. And that will help us think about how to reverse engineer, if you will, a vaccine. If we know what the immune response is that you need to have and how much antibody you need to have in you in order to successfully fight off an HIV infection, that'll help us sort of work backwards and work to develop vaccines that would be able to elicit that response. We also think that it'll help us look at the kinds of models for lab tests and animal studies that we do to design models that can be more effective in predicting human outcomes. So we have a lot of data about this antibody from labs and from studies in non-human primates. And if we find that this antibody works, and for that matter, if we find out that it doesn't work, we'll be able to go back and look at those data and try to figure out how could we have designed a better experiment that would have been a better predictor of responses seen in people, so that going forward we can be more efficient and do all of that early stage work in ways that are more effective, potentially saving a lot of dollars, obviously, but then also help us get into clinical trials with a better set of information so that we have more information that's truly useful to understanding a human response. Next slide, please. So, and that last point is really kind of the key factor. We have not been successful yet in finding a vaccine that has been able to successfully get the human body to produce these neutralizing antibodies in enough quantity that they could be protective. The closest we got was the Thai trial, RV144, which was only about 30% effective, or 32% effective, excuse me, at reducing new infections. And while that's exciting and promising, and there's a whole lot of upcoming work going on to build on that result and see if we can improve on that outcome, so far we're not there yet. So we need to know how much neutralizing antibody is enough to achieve protection, and we need to understand how to use these models to get us better data. And we think that the bottom line there is that by answering those questions, we'll be able to move vaccine research broadly forward in a more effective way, and certainly in a money-saving way. Next slide, please. So how could we use this technology as a prevention tool? And I mentioned this earlier a little bit. So we think that it might be possible to use in infants who are born to HIV-positive moms to protect them both during pregnancy and during delivery, but also after delivery and during breastfeeding. We think that they might have the potential to serve as kind of a backup to using long-acting PrEP given by injection. So if you can imagine for a minute, if you get an injection that you know is good for a year, it's reasonable to assume that early on in that year is when that injection is probably the most potent and is working at its optimal strength. And the longer amount of time goes by in that year, the amount that remains in your body is waning over time. So there's the potential, if this works, that people could get an infusion to help cover that sort of tail end period of time when the coverage of that injection of PrEP is waning. And obviously we need to know more about how those long-acting injections work as well. There's other trials being done by the HPTN to move that research forward. So this has some potential to work in collaboration. It could also potentially work on the front end. If we have vaccines, for example, that have to be given over a 6-month period of time or a 1-year period of time before they're fully effective, maybe this is something that people could use in combination during that initial ramping up phase of giving a vaccine regimen. And then potentially maybe this is something that could be given with other antibodies in some sort of a cocktail, if you will, that maybe one individual antibody wouldn't be sufficient to cover the broadest global strains of HIV, but we know that there are a number of other antibodies that have been discovered as well by a number of different research teams. So maybe there would be the potential to use them in some sort of combination mixture to get the best coverage of all the different global strains of HIV. So again, these are all hypotheses. None of these are things that actually are able to be done in real-world scenarios right now, but this is the sort of thinking behind the AMP study and how we envision the potential for having this kind of a tool available to us. Next, please. So this little video is just a way to sort of show you in animation what's actually happening with these antibodies. So down at the bottom there, that host cell, that's you, that's a T cell. And those little green Y-shaped antibodies would be present in your system from the infusion so that if HIV comes along, they could attach on and block HIV. And Matthew, if you'll run that one more time if it doesn't repeat automatically. Oh, there it goes. So our T cells have these little receptors on them. That's where ordinarily HIV would latch on to cause an infection. We'll give you these antibodies. You'll have them floating around. HIV comes along. They bind on to the protein spikes. They block those protein spikes, and you can see that the protein spikes would not be able to attach to the T cell to cause an infection. So just a little quick animation. I'll let you watch it once more without me chatting so you can just take it in. Great. Next slide, please, Matthew. So what are the research questions in the AMP study? There are actually 4. As we always do, we want to continue to look at the safety profile. And when we're working in a larger population, we have the ability to detect a greater range of safety data. We also want to continue to look at the question of tolerability. Are people able to take this antibody through an infusion without being too uncomfortable? Are they able to tolerate the procedure without any kind of negative side effects? Those are similar to the questions that have already been looked at in the phase 1 early stage research. But then unique to this advanced study, we want to look specifically at, does this antibody lower the likelihood that people would get infected with HIV? Will it prevent infection? And if in fact it does, what is the level of antibody that is needed to provide protection? So next slide, please. In order to join the study, people are going to go through a screening process to determine if they're eligible. That includes sort of 2 big domains. The first domain is on the biology side and people's health. So we're going to assess that through doing a complete physical exam, looking at the usual kinds of things like weight, blood pressure, heart health, their temperature, do their kidneys seem to be working well when we look at urine samples and things like that. We're also going to be doing HIV testing because this is a study that will only enroll people who are HIV uninfected. And then the second big domain of that screening process is to look at people's behaviors and what their risk factors are for HIV. This is a study that will enroll people who are at risk. So we will be looking closely at the kinds of sexual behaviors that people describe that they engage in, and any kind of alcohol or drug use profiles that would contribute to them being at risk. And as we always say, there are any number of factors that could cause someone to be not eligible, And we work, if we identify any kinds of health problems that people might not have been aware of, we help them to access care and provide referrals as needed to help them get whatever sort of medical attention they need for any issues that have been identified. And if we identify through HIV testing that people are in fact already infected and just didn't know, we'll be working with, each site will work in their communities to refer them to care, help engage them in the care continuum, and get them access to the services that they need. Next, please. So the three groups of the study, are going to look at different doses compared against a placebo. In this study, our placebo is going to be normal saline solution, or what I think lay people commonly think of as just sterile salt water. This is the same kind of saline solution. If any of you wear contact lenses, it's the same kind of saline solution that you're putting in your eyeballs. And then the two intervention arms in the study are looking at two different doses of the antibody. There'll be a low dose at 10 milligrams per kilogram and a high dose at 30 milligrams per kilogram. And that dosing information is a little bit unique. That per kilogram piece is a little different than anything you might have seen previously. And that's because we want to give the antibody relative to a person's weight, so that whether you're a person who weighs 100 pounds or a person who weighs 200 pounds, you're getting the same amount of antibody per pound regardless of your weight. So part of our study procedures will be to weigh participants and then calibrate their dosage based on that weight. And again, the idea of testing these two different doses is to help us get at the question of if in fact there is protection seen, how much antibody do people need to have on board in order to achieve protection. So that earlier slide that I showed you, the lab data, we were looking at doses in micrograms, so comparatively much, much smaller because they were only being used in a blood sample in a petri dish or in a test tube. And so now we're looking at a comparable dosage that's been adjusted for the size of the human body, and then our low dose and high dose are calculated again based on the individual volunteer's weight. Next, please. So how do we decide who goes in which group and who gets which product, or which dosage rather? And again, this is the idea of randomization. So like tossing dice or like flipping a coin, we are going to randomize people to different groups in the study. Two of the groups are going to get the placebo, two of the groups are going to get the low dose, and two of the groups are going to get the high dose. And I think, Matthew, if you'll advance, I think the next slide shows the schema. Yep. So to show you why there's those two groups for every dose and so on, that's because we've got these two different cohorts. So one group will be the women in Africa, and one group will be the MSM and transgender individuals in the Americas. The sizes of the groups vary a little bit just because of incidence and prevalence in the countries. So the American and South American groups need to be a little bit bigger than the Sub-Saharan African groups. But again, equal distribution across the three groups, so we'll have even comparisons. And all groups are going to get the same number of doses, a total of 10 infusions given every 8 weeks. And I'll be showing you more about that in just a minute. Next slide, please. And as is typical in a randomly controlled trial, the whole idea here is that we do the same thing in every group, and the only thing that varies is which product assignment people are getting. So everybody is going to be receiving risk reduction counseling and the best available risk reduction package, helping people access PrEP if they're interested in taking it, and I'll say more about that in a minute. We're going to be following everyone for 22 months, checking their immune responses, checking their HIV status monthly, And then at the end of the study, we add up the numbers, and it's literally as simple as doing the math and comparing the numbers of infections that may occur in any of the three groups and determining whether, in fact, those are statistically significant and whether that antibody was protective. Next, please. So again, PrEP. How are we going to incorporate that in this study? So it's going to vary a little bit by region, and that's because of the differences in licensure in different parts of the world. In the U.S., where PrEP is licensed, PrEP will be incorporated into our risk reduction package. If people are already using PrEP, that does not change their eligibility for the study. If they're not currently using it, but they're interested in using it, our sites will be working with them to refer them to a PrEP provider to write the prescription and follow all of the monitoring and blood testing and so forth that needs to happen for people using the drug. And we have arranged a program with Gilead to provide the drug itself at no cost. So the only cost to the individual is if they have any sort of study, office visit copays, if there are labs that need to be run, those kinds of costs. But the drug itself is provided free. In South America, there are demonstration projects available, and any interested participants will be referred to those demonstration projects. They can participate in our study and the demonstration project at the same time. There's not any conflict there. And in Africa, there is not yet any African country that has licensed PrEP, and so far only South Africa has a licensure application under review. So for the moment, PrEP is not being provided to our African participants. However, we expect that the status of those licensure applications will change, and so we expect that the study will change to match. And as any additional licensure applications or demonstration projects become available in Africa, the study will be modified to match and making sure that we can provide the same level of access as in the U.S. and South America. Next, please. I think a concept that's probably familiar to everybody on this study is that on this call rather, is that the study is double-blinded. So the participants will not know the contents of the IV bag. It will just look like clear liquid, and they won't know whether it's got antibodies in it or not. Only the pharmacist at each of the clinics will know those assignments, and pharmacists do not interact directly with participants, so the blinding is maintained. Next, please. Just to give you a quick idea of the kinds of things that are going to happen at study visits, I mentioned we're going to be doing HIV counseling and testing every month. There's periodic physical exams to help check people's safety and make sure that they continue to be in good health. Collecting blood and urine samples is part of that. At a couple of points in time during the study, we collect swabs to look for other kinds of sexually transmitted infections. Anyone who is capable of becoming pregnant will be tested for pregnancy. We'll also be collecting data about medicines that people take, including whether they use PEP or PrEP, and we'll be tracking antiretroviral blood levels in the study as well, so that we can better understand how the antibody may be interacting with the medications that people use. And we'll also be checking any kinds of problems or benefits that people are experiencing as a study participant. Next. So I mentioned the number of infusions and the study duration. So this is for each individual participant. They will be enrolled for 22 months or just under 2 years. They will be getting an infusion at only 10 of those visits every 8 weeks. Which is roughly every other month. But the study visits are scheduled to occur every 4 weeks. So we'll be doing monthly HIV testing, and then at every other visit for those first period of time they'll get the infusion. There is a period of time at the end of the study after they've had the last infusion, where we can continue to monitor their health for a couple of months after that infusion series is done. Next, please. We'll also be in close contact with the participants directly following each infusion for 3 days. So we're looking for any kinds of side effects they might experience. So just as examples, headaches, do they feel weak? Are they experiencing any sort of rash or itching? Do they feel tired or run down? Do they have a fever? But those are just examples. The clinic staff will be working with participants to monitor any kinds of reactions they experience, and they'll be following them for 3 days after they receive the infusion to track that information. Next, please. So again, just kind of recapping the main responsibilities for our study participants. They'll be getting the IVs, which takes between 30 and 60 minutes, and that happens every 8 weeks. They'll be getting blood draws every month for checking immune responses and HIV testing. I mentioned we'll be doing the STI testing with swabs. That happens about every 6 months. And then the one thing I didn't mention previously, there'll be a number of different questionnaires that get completed every 4 to 8 weeks, looking both at general health questions and also keeping track of people's sexual behaviors, trying to understand if anybody's behavior is changing as a result of being in the study, and to help contribute to providing good risk reduction counseling. Next, please. We often get a lot of questions about what do we do with the blood that we take. So I've got a couple of slides here to help address that domain of information. So at our study visits, we're going to be taking between 10 milliliters and 160 milliliters. And since most of us are not metrically minded and might be more familiar with things we would find in our own kitchens, you can think of that as 2 teaspoons to 2/3 of a measuring cup. So not terribly large amounts of blood, and in terms of, if you're familiar with the sort of average size of the tubes that blood is collected in, that's between 1 and 16 tubes at any given visit. Next, please. What do we use the blood for? So obviously we're using some of it for the HIV diagnostic tests. We're also using some of it for monitoring of health, things like a CBC, which measures the different components of the blood, like red cells and white cells and such. Also using some for tests that look at liver and kidney health function, so ALT and creatinine tests. And then the bulk of it is stored for tests that are done in the future. And on the next slide, we detail those a bit. Those are tests that look at immune responses, and also tests that look at the level of antibody that's present, sort of repeating some of the earlier lab tests to look at how the VRC01 is neutralizing if we were to take that blood sample and spike it with HIV to see if neutralization is happening. I mentioned we're going to be looking to detect whether or not people are using ARVs, like PEP or PrEP. And the kinds of immune system testing that we'll be doing is looking at how the body reacts. So things that are unique to individual based on their genetic makeup, how the other pieces of the immune system might react to the antibody, and also in people who do become infected, doing some viral sequencing to understand why that particular HIV strain seemed to not respond to the VRC01 and why neutralization did not occur. And we can do that, or hope to be able to do that, by looking more carefully at the strains that people do get infected with if infection occurs. Next, please. We also get a lot of questions about how being a participant impacts HIV testing. And for any of you that have been around for a while and have heard about vaccine trials in the past, you may have heard about something called VISP, or vaccine-induced seropositivity. And this is the idea that if you're using the common kinds of rapid tests for a diagnostic that look for the presence of an antibody as a proxy for HIV, people who are in a vaccine trial where the vaccine is designed to help you make antibodies may have positive responses on those tests and therefore could be mistakenly identified as being HIV infected when in fact they may be fully uninfected and just responding to the vaccine. You'll be glad to know that that is not an issue in this study. This antibody is not detected by any of the common antibody tests that we've been able to look at. We've tested against a whole lot of test kits, not only in the US, but test kits from around the world. And the thinking is that the test kits are looking for reactions that would be a sign of infection that happen quickly and happen commonly, because obviously you want to have a test that works in the greatest number of people possible and that can work to identify an infection very fast. This antibody is neither of those things. It doesn't develop quickly. In the individual that it was originally found in, that was a person who had been HIV infected for over 10 years at the point in time when the antibody was found. So this is a rare, and it's also a rare antibody. So it's only being identified in the, that very special class of some people who are considered elite controllers, not even all elite controllers. So it's a reaction that is slow and rare, whereas antibody diagnostic tests are designed to look for things that are common and fast. So to the best of our knowledge at this point in time, we do not have any concerns about HIV testing. People won't have to be worried about that. We are going to recommend that they continue to get their HIV testing done only at our research sites, and that's largely just out of an abundance of caution, because HIV testing technologies do change. They're constantly evolving. We've been hearing about fourth-generation tests that may look for the presence of an antigen and not just the presence of antibodies. And we don't know how those tests will work in reaction with the antibody. So we are going to encourage our study volunteers to be tested only at the study site, but really that's out of an abundance of caution and for no other reason. But once the study is over, they are welcome to resume whatever their normal HIV testing practices were, and we don't anticipate that that's going to pose a problem for anyone. Next, please. Almost done. In fact, I am done. That is the last slide. So we'd love to open it up for questions. I do want to acknowledge all of our partners in putting together these slides and in working on this study. My counterparts at the HPTN, the rest of my community team here at the HVTN, our lab partners, our protocol team, and of course our funders at the National Institute of Allergy and Infectious Disease who are making all of this possible. And I think on the last slide there's just my contact information for anybody who wants to follow up in the future, but we'd like to open it up for questions now.
So I'm Miriam Jones. I just unmuted your line. You can ask your question.
Hi, Miriam, are you there?
Oh, wait, Josh, you have a question?
Yes, I have a question.
Hi.
Okay, so I noticed that participants have to come back every, is it 8 weeks to get re-injected, I guess, with the antibodies, is that right?
Reinfused, yes, that's correct.
Okay, so after that 8-week period, each time, like, do the antibodies go away, and after they're done with the study, after a while, do the antibodies in their blood go away, or are some still present?
You are exactly correct. So people have to get an infusion every 8 weeks because these antibodies that are manufactured in a lab don't last long the way naturally produced ones would. So, and that is why we have those extra couple of months at the end of the study after they get their last infusion. It's also one of the reasons why we're not worried about HIV testing after the study's over. because by the end of the study, we would not expect that people would have any antibody left in their system. You're exactly right.
Okay. Can you still hear me?
Yep.
Okay. So, okay, within that period, okay, it's you come back every 8 weeks. So does it actually last a little longer than 8 weeks, or is it cut off at that 8 weeks and then you have to immediately come back?
There's always this issue of kind of there being a tail, and you'll probably hear about this as you guys have future conversations about injectable PrEP that's long-acting as well. So the amount is diminishing over time, and the 8-week mark is thought to be sort of the point in time where that tail is getting small enough that you wouldn't expect it to be protective, and so that's why that's the time point to give the next infusion. But there is a little bit of wiggle room, if you will, in that we've seen different kinds of graphs and stuff to show kind of how these doses taper off over time, and there's a little bit of forgiveness in there.
Okay, I think I have one more question, then I'll leave you alone.
Sure.
Okay, so what kind of lab tests did you guys do to say, okay, this antibody worked? Like, did y'all culture HIV with cells or something? Like, how did that even come about?
Yeah, so I mean, the beauty of the program is that we can use these blood samples that we take from our volunteers, and we can put them in test tubes and petri dishes and do all kinds of experiments that don't put our volunteers in harm's way.
Okay.
So, you know, we can take a blood sample and put it in a test tube and, you know, spike it with HIV and then, you know, see how it reacts for things. So we know that in doing it that way, we can protect the safety of our volunteers, but we can also answer the scientific questions.
Okay. We're going to go to Miriam. Are you there? Miriam, no? Okay, questions from the queue. Have all the participants been selected for the trial, all 800?
No, none of them have been yet. So we are not actually open yet. We are waiting to resolve the last final questions that have come from the FDA, which is a typical part of the regulatory review process. So we're working out those last few questions that the FDA had for us and resolving those to their satisfaction. We're expecting that the study will open in the U.S. in the first quarter of 2016, and that is when our studies will begin their active efforts to identify participants or identify people who would be interested in participating and start doing the screenings to find people who are eligible. So that has not started at all yet.
Okay. The next question is, where would the study be conducted, and are there any ASOs available for those who are not adherent to the infusion trial, in which may be newly infected for referral services to HIV care? Right.
So, Matthew, I don't know if you can flip back to that early slide that showed the cities in the U.S. So we have a total of 19 clinics in the U.S. that will be participating. And they are all very closely connected to the programs in their cities to be, as you said, that as people become HIV infected, we want to link them into care as quickly as possible. So our studies are familiar with the services and providers available in their areas. They do link people up, whether it's AIDS service organizations, whether it's healthcare service providers, whatever it is, and it's different in every city. So our studies work very closely with the other providers in their communities to make sure that those volunteers who are identified as being infected do get referred into care. And as you noted, you know, people may not be compliant with the prevention methods that they're using, so there may be people who become infected, or you know, people may drop out of the study at some point, for example. So we want to make sure that we connect them up with the services that they need, and our clinics do a great job of that.
Okay, and I can answer the last question in the queue, which is, are there any additional clinics and sites being included to expand the reach of the study? I will say that at this point, no, as the study hasn't started to enroll yet.
You're exactly right. If we find that there are delays in enrollment, or that the capacity that we have with the sites that have already been identified is in some way insufficient, then absolutely we would look at bringing on additional sites in the future. But since we haven't started yet, we're not at that point yet.
Well, if there are any other questions, going once, going twice, if there are, please email me. I want to thank Gail for being on the line and giving this fabulous presentation. Like I told you in the start, it is great and interesting science and really important for folks to know where we're looking, where we might be going, and how our community should understand what's happening. So I thank everyone for joining us, and till next time, thanks again, Gail.
Thanks for having me. Much appreciated.